Welcome friends. Let's continue our discussion about the cell. We are studying basic aspects of the cell. In our previous videos we have discussed the structure of the cell, all the cell organelles which are present in the cell, types of cell, size of the cell, function of the cell, cell organelles, and lastly about the nucleus. Now let's discuss about one of the important aspect of the cell, and that is cell division. The cell division basically is mitosis and meiosis, two types. One cell gives rise to two cells, two daughter cells in mitosis, and one cell gives rise to four haploid cells where there is a reduction in the chromosome number, and that is the case in meiosis. In today's video we will discuss about the mitosis, a cell division in which there is no reduction at all. So we will discuss today with the help of some. In case of the mitosis, again before going to mitosis, let me explain that the cell division might be amitotic also, and we know that there are certain bacteria and few other organisms like amoeba, yeast, they show amitotic cell division where the mitosis is not followed, the steps in the mitosis are not followed. In this type of cell division, which is we call it as binary fission, it was first described by Remark in 1841. It doesn't follow the cell cycle. The mitosis is a cell division which is taking place according to the cell cycle. But in case of the mitosis, there is no cell cycle. In case of the amitosis, the cell and the nucleus or DNA, if it is a prokaryote, it is only DNA in place of nucleus. So both nucleus and the cell or DNA and the cell, they elongate and they constrict in the middle. The cell constricts in the middle and it splits off into two daughter cells. So just very simple bifurcation of the cell. One cell divides into two cells without much complexity. Now this occurs in, as I told you, bacteria, amoeba, yeast and few other organelles. Now let's come to the typical cell cycle which is followed in the higher organisms and in mitosis. Mitosis is a part of the cell cycle. The cell cycle was first described by Howard and Pelk in 1953. It is a series of programmed cyclic changes by which cell duplicates its content and divides into two parts. So a cell undergoes a cyclic changes, series of cyclic changes. These are programmed, these are particular, they are followed in a particular manner, and lastly the cell divides into two parts. So that is known as a cell cycle. It is divided into, cell cycle is divided into two phases. The first phase is interphase. It is non-dividing phase. The cell is not dividing in interphase. It is preparing itself for the division. So before undergoing cell division, the cell needs a preparation, and that preparatory phase is the interphase. This interphase is again divided into G1 phase, S phase and G2 phase. And the second phase of the cell cycle is the mitosis, mitotic phase. It is the dividing phase and it is divided into prophase, metaphase, anaphase and telophase. Now coming to the interphase, a newly formed cell enters into the interphase. When the cell is formed at once, it enters into the, so after completion of the division, the cells enter into interphase, and before cell division also they are in the interphase. So interphase is in between the divisions, two divisions. So a newly formed cell, when enters into interphase, it goes through many changes. It becomes ready for cell division. It involves a replication of DNA in interphase. The DNA is replicated. One DNA undergoes replication and forms two DNAs. It is synthesis of nuclear proteins, whatever nuclear proteins, histone proteins which are required for binding this DNA to form the chromatin or chromosome, they are synthesized here, and the centrosome is also duplicated because each cell needs centrosome. When the cell is going to divide, it needs its own centrosome. So centrosome is also divided. Interphase is divided into three stages: the G1 phase, S phase, and the G2 phase. G1 phase, also called as gap phase 1 or first gap phase, it is a resting phase comparatively, but even then some instances or some synthesis takes place here also. The cell enlarges in size, synthesis of RNA, some carbohydrates, some proteins, and some lipids, they are synthesized in this G1 phase, gap 1 phase. Then S phase is a typical synthetic phase. Here the DNA replication takes place. That is the most important thing. One DNA after replication becomes two DNA. So one DNA becomes double. It is actually duplication of the DNA or replication. Then centriole replication. Two centrioles are formed from one. Z2 phase is the third phase in interphase, and this is a gap phase 2, or also called as second gap phase. It is again a resting phase. Comparatively, it is resting as compared to synthetic phase. In synthetic phase, the cell is very busy. DNA replication, centriole duplication. So it is very busy in S phase. Comparatively, in G1 and G2 phase, the cell is not that much busy. Even then, DNA repair, if there is some problem, because DNA has replicated in the S phase, so its repairing is done in G2 phase. Synthesis of the RNA, some RNAs are synthesized in G1 phase also. Here also some of them are synthesized RNA, and duplication of the cell organelles. Whatever cell organelles like ribosomes, they are present at the time of cell division, so they are duplicated here because the cell is going after G2 phase, the cell is entering the division phase, and when one cell divides into two cells, each cell needs its own cell organelles. So cell organelle duplication is completed in G2 phase. Now after G2 phase, the cell is ready for division. Before that, let's see the diagram here. Interphase, it is divided into, I will show with pen here, laser. So this is the G1 phase, then S phase, then G2 phase, and this is the division, cell division. This is karyokinesis, when there is division of the nucleus, and this is cytokinesis, division of the cytoplasm. After that, two cells are formed. Again, the cell which is formed here, it enters into interphase. Initially it takes some gap or some RNA proteins, they are synthesized. Then S phase, the cell comes into the S phase where DNA is doubled. Then again it enters into G2 phase where some RNAs are synthesized, DNA is repaired, and then it enters into the divisional phase. So in this way, each cell completes its cell cycle in this way. If we see the time period, here we can see the time period. In G1 phase, the cell may remain up to 12 hours. Then in S phase, synthetic phase, 6 to 8 hours are required, and then in G2 phase, 3 to 4 hours. The cell completes its division in just 1 hour. Again, it takes a small gap here, G0, then enters the G1 phase. So this is time wise: 12 hours in G1 phase, 6 to 8 in S phase, 3 to 4 hours in G2 phase, and 1 hour in mitotic phase, divisional phase. Now come to the mitosis, typical mitosis. It is a process of cell division which includes karyokinesis and cytokinesis. Karyokinesis is the division of nucleus, and cytokinesis is the division of cytoplasm. The karyokinesis is completed into 4 steps in mitosis. mitosis. Meiosis is different. In mitosis, it is completed into prophase, metaphase, anaphase, and telophase. 4 phases are there. Let's see one by one. First one is the prophase. In case of the prophase, the chromatin fibers condense. Before entering the prophase, we know the cell is interphase. So in interphase, the typical nucleus is present with a nuclear membrane, nuclear envelope, nucleolus, and chromatin fiber. So chromatin, now we would call it after condensation, we would call it as chromosome. Before condensation, it is chromatin, thin filaments of chromosome bounded with some histone proteins. They are present in interphase. Now they start, the chromatin material or chromatin fibers, they start condensing to form the chromosome. The chromosome shortens, it becomes distinct, each with two chromatids attached at the centromere. Here you can see the chromosomes appear now in the prophase. In the early prophase, chromatin is present and it starts condensation. In the late prophase, chromosomes appear with centromere. Each chromosome is having two chromatids. The centrosomes reach opposite poles. Initially, both the centrosomes lie near one another, but now they reach to their poles. It goes here, it goes here. So the centrosomes reach the opposite poles and they start forming the spindle fibers. These fibers are the spindle fibers. Half spindle is formed by each centrosome. The nucleolus and nuclear membrane disappear. Here the figure is showing nuclear envelope, but it is disintegrating now. Its disintegration has been started. Disorganized, the nuclear envelope and this nucleolus both are disorganized towards the late prophase. Here this is the interphase. Now in the interphase we don't see chromosomes here. Chromatin is present and it is distributed throughout the nucleus. So when we apply a nuclear stain, specific stain for the chromatin, the whole nucleus is colorful. It becomes colorful. It is stained because the chromatin is spreading throughout the nucleus. This is nucleolus, very clear here. But in late prophase, or we call it as pro-metaphase, just prior to metaphase, it is pro-metaphase. In the pro-metaphase, you can see the chromosomes at the most condensed phase. They show clear-cut two chromatids, kinetochores, and the nuclear envelope. These are the pieces of nuclear envelope you see here. The nuclear envelope is disorganized, this nucleolus is disorganized, the centrosomes are reached, they have been reached to their respective poles and spindle is formed here. Now this is pro-metaphase. Now come to the typical metaphase. The metaphase, identity of metaphase is the chromosomes arranged at the equatorial plate. So this is the equatorial plate or the cell plate, and at the center they have been arranged. The chromosomes get attached to the spindle fibers. Now here you can see this is a double spindle fiber attached to the kinetochore here, and in this way the chromosomes are attached to the spindle fibers. All the chromosomes lie at the equatorial plate, forming a metaphasic plate. This is the equator and it is also called as metaphasic plate. So all the chromosomes lie at the metaphase. They are not distributed throughout the cell here. Now the nuclear envelope is absent, it is disorganized. The whole cell is available for the chromosomes, but these centrosomes, these microtubules of the spindle, they arrange these chromosomes at the center at the metaphasic plate. The chromosomes at metaphase are the shortest and thickest chromosomes. Chromosomes at the metaphase, they are the shortest chromosomes, they are the thickest chromosomes. Now coming to the, I would try to explain more about the spindle formation. So here you can see metaphase, all the chromosomes are at the center, they are arranged at the equatorial plate, and here you can see centrosome with two centrioles, a pair of centrioles. It is the half spindle. They the spindles elongate and they get attached to the chromosome. So this is a chromosome, this is a chromosome with two chromatids. This is a chromatid, this is another chromatid. So in each chromosome two sister chromatids are present, and here you can see a green colored spot, that is the kinetochore. So a spindle comes and attaches itself to this kinetochore. The spindles are attached to the kinetochore. You can see two chromosomes here with four chromatids, and the spindles are attached. One spindle from opposite poles. It comes from this centrosome, this comes from this centrosome. Again, it is seen very clearly in this figure. You see, this is the two centrioles in the centrosome, and this is the spindle fiber which is attached to the kinetochore. Now the spindle which is attached to the kinetochore, or the microtubule which is attached to the kinetochore, is known as kinetochore microtubule. So this, you can see this line, this is a kinetochore microtubule, this is a kinetochore microtubule, this is a kinetochore microtubule, this is again a kinetochore microtubule. These are the microtubules which are attached to the kinetochore of the chromosome. So this is the metaphasic structure at metaphase. In this way the spindle is formed. And now remember that in case of the animals, the spindle is formed by these centrosomes. Centrosomes are present only in animals. In plant cells the centrosomes are not formed. In plant cells, the centrosomes or centrioles are absent, but even then the spindles are formed, and these spindles in plant cells, they are formed by the microtubules. The microtubules are made up of the tubulin protein. So monomers of the tubulin protein, like this, these are the monomers of tubulin protein, they get attached to one another and the spindles are formed. So spindles or microtubules are formed in the plant cells, in the animal cells, in both cases. In animal cells the centriole is absent, centrosome is absent, the spindles are formed by the tubulin protein, and in case of the animal cell, the centrosome is forming the spindles. So this is also called as aster. So aster formation is the case in animal cells. Aster is not formed in the plant cells, but the tubules are formed, the spindles are formed, and they pull. Now here you can see they pull the chromosomes to their respective poles. Now when in case of the anaphase, now before that come to the metaphase here. Now this is a chromosome, you can see this is a chromosome with two chromatids or sister chromatids. Now this chromosome is splitting at the centromeric position. So here, now this is, here the centromere is present. So this centromere splits, one of the chromatid is pulled to this pole and this would be pulled to another pole by the spindle. In this way, in anaphase further, here you can see the pulling of the chromatids. Now these chromatids are called as the daughter chromosomes. These are called as the daughter chromosomes. You can see here in the figure, daughter chromosome. So this chromosome, it is actually a chromatid of the double chromosome. So it is pulled to this pole and this is being pulled to this pole. And when they are separated in this way, the anaphase begins. So anaphase is a stage in which the centrosomes of each chromosome divide and they form the daughter chromosomes. The sister chromatids are now called as daughter chromosomes. Spindle fibers start pulling these daughter chromosomes to their respective poles. Now coming to the, now the anaphase is a stage where the pulling of the daughter chromosomes is seen. When they reach to their respective poles, again they uncoil and they the cell or the stage is now called as telophase. So in a telophase, again nucleus formation takes place. When the chromosomes are pulled to their respective poles, again the nuclear envelope, you can see here the nuclear envelope formation begins, the spindles are cut, and the chromosomes which were at the thickest level in the metaphase and anaphase, now they are relaxed. Their length increases and the thickness decreases. So the daughter chromosomes uncoil, they elongate and they form chromatin fibers again in the telophase. Nucleolus and nuclear envelope, they reappears forming two daughter nuclei. Here this would form one nucleus here and this would form another nucleus here. In this way the two nuclei are formed at the end of telophase. In animal cells, the cytokinesis takes place by cleavage of there is a difference in plant cells and animal cells. We will see here the difference. So this is a case in plant cells and this is a case in plant cells. Sorry, this is the first one is the case in animals. So this is a animal cell and this is a plant cell. In case of the animal cells, there is cleavage. You can see here the cleavage. When the telophase or after anaphase, when the chromosomes are being pulled to their respective pole, at the same time a cleavage is formed in the plasma membrane of the cell in case of the animals. So in animals the cytokinase is completed with telophase by Furrer method, a cleavage method or Furrer method, and it is formed from outside. It begins from outside and it goes towards the center. The cleavage continues from both the sides and lastly it makes in the center, and in this way you can see here the two cells get separated. Now this cleavage formation has begun in the late anaphase, and with telophase, completion of the telophase, when two separate nuclei are formed, at the same time the cytokinesis is also completed. Cytokinesis is the division of cytoplasm. So nuclear division is followed with cytoplasmic division, and in case of the animal cells it is done with the help of cleavage formation or the Furrer method. In case of the plants, we know the cell wall is present. So this is the cell wall, and therefore as cell wall is present in the plants, the cleavage formation is not taking place here. Now what happens, this is called as a cell plate method. In this cell plate, at the center of this cell you can see such type of small vesicles are arranged. Now these vesicles are cut by the Golgi complex and the endoplasmic reticulum. So these are the membrane bound small vesicles, and these vesicles are cut, they are cut by the Golgi complex and the endoplasmic reticulum, and they get arranged here at the center of the cell in between these two nuclei. Now naturally in case of the animal cells, the here in case of the animal cells, after anaphase the cytokinesis starts, but in case of the plant cells the cytokinesis begins after completion of the telophase. When two clear nuclei are formed here, then the cell plate formation begins, then the cell wall at the center is formed. Now these are the membrane bound vesicles, they are forming the cell plate. Here is the cell plate. In the cell plate, these vesicles, they are attached to one another and they continue, two layers are formed. One layer is on this side, another layer is on this side. So two layers are formed here. Now these two layers become the layers plasma membrane of the cell in this way. They become the part of the cell in this way, and at the center of these two layers the middle lamella is formed. So this at the center, it is middle lamella, and these two cells are now separate with their separate plasma membrane and cell wall. They have their own plasma membrane, they have their own cell wall, and in between these two, here the uh, middle lamella is present. So in this way, by means of the cell plate formation, the plant cells divide. The cytokinesis in the plant cells is completed by means of the cell plate formation. There is now basically you can see in case of the animal and plant mitosis there are two major differences. The first difference is of the ester formation. In case of the animal cells, the centrioles are present, centrosome is present, and therefore ester formation takes place. In case of the plant cells, ester formation is not taking place because the centrioles and centrosomes are absent in the plant cell. And the second difference is at the time of cytokinesis. In cytokinesis in animal cells, the cytokinesis is by the Furrow method or cleavage is there in the plasma membrane, and in case of the plant cells, the cell is divided or cytokinesis is taking place by cell plate formation. Now again there is difference because here the in case of the animals the cell is divided or the cytokinesis is from outside to inside. You can see here from outside to inside, and in case of the plants it is from inside to outside. The cell plate is begun at the center and then it is elongated towards the poles, towards the periphery. So the cytokinesis in the animal cells is from outside to inside, and the cytokinesis in case of the plant cells is from inside to outside. Now this is the cell cycle complete, again mitosis, not whole cell cycle. Here you can see this is newly formed cell, it is in interphase. Now in interphase here you can see the centriole is doubled in S phase. In S phase the chromosomes are also doubled. So here the chromatin is present and this chromatin is having a single like this, a single chromatid in it, and here it is doubled. Two chromatins are present in each chromosome like this. So it is doubled here. So this happens in the S phase, centriole duplication and the chromosome replication. Now this is the part of S phase. Then in interphase, then it enters into the prophase and into metaphase, into anaphase, then this is metaphase typical, this is late and prophase or early metaphase. So prometaphase, this is prometaphase, this is metaphase typical, then anaphase, then this is again anaphase, this is telophase where the two cells are undergoing cleavage and cytokinesis. Two complete cells are formed here in this way. Now this cell again enters into the cytoplasm division if it wants to divide, otherwise it becomes mature cell. Cell maturation takes place and it becomes a it is adhered or it is given a particular function and it becomes a permanent cell. So either it becomes permanent cell or it becomes the it undergoes again the divisional phase in this way, cell cycle. So coming to the significance of mitosis now, mitosis is one of the most important cell division and it is important because it is resulting in the growth. Now in case of us human beings, we know that in mother's womb we are starting our journey from a single cell, fertilized cell, which is known as zygote. So that single cell zygote develops into our body and our body is made up of some trillion cells. Now this increase in the cell number is the result of mitosis. So out our life cycle we are continuously showing the mitotic cell division. So development of the organs, development of the organism, body of the organism or growth, anything, all this is the result of mitosis. Without mitosis we all would have been single celled animals. We are multicellular organisms because we have the capacity to divide by mitotic cell division. In our body, we always clear our body, and old, decaying or dead cells they are removed and new cells are added continuously. Even at a particular phase of the life, we know that our growth is stopped, but cell division is not stopped. Mitosis is not stopped. Mitosis is a continuous process because the older cells or dead cells they are replaced every day by new cells, and the new cells are added by mitosis. In many organisms, asexual reproduction result in mitosis. Asexual reproduction. It is the result of mitosis. Whenever budding is there, grafting, all these asexual means gall body formation or the bulbil formation, whatever methods are there, amitotic, asexual methods, all of them are the result of mitosis. The gonads and sex cells increase in number with the help of mitosis. Whenever the number needs to be increased, even after meiosis, suppose sperms are formed, suppose eggs are formed, and they need to be increased in number, mitosis takes place. So whenever the number, it might be of gonads or ovaries or testes or sperms or any type of a reproductive structure, if its number needs to be increased, it is increased with the help of mitosis. Mitosis helps the cell in maintaining the size. When the cell increases, it undergoes mitosis and it maintains its size and shape also. So in this way, mitosis is one of the most important cell division. I think it is sufficient for today. Thank you.